Hello everybody, today's class is about DNA replication. So if you remember well, when we looked at the cell cycle, right, we know that the cell cycle is like this. We have G1, then we have the S phase, then we have G2, and then we have a part here that is called mitosis. And in mitosis, the cells, so from one cell here, at the end of this cycle here, at the end of mitosis and everything, then we end up with two cells. Okay, so we end up with two cells, and these two cells will now be going back into a cell cycle of their own. So, and then, so the cells will go back to G1, etc., right? So we know that most cells spend most of their time in G1, and then eventually, when it's time to go to create two daughter cells, those cells will go to the S phase, G2 and M, and then merge and then divide into two cells. Okay, so we know that. We've looked at this. We looked at the phases of mitosis and everything. Now, what we want to do at this point for this video is basically concentrate on the S phase. Not the S phase per se, but actually looking at what the cell does during this S phase. And in that case, what it does is that at the beginning of the S phase, just as the cell is just about to enter the S phase, our cells have, so I'm talking about, say, human cells, just as an example, but you can imagine that any diploid organism goes through this. So we have two N chromosomes, right? So in our case, 23 pairs of chromosomes, total of 46. One pair inherited from your father, the other pair inherited from your mother. 23, 23, the gametes come together, there's fertilization, a zygote is made, and then you are now a diploid organism, and that organism has two N chromosomes, in our case, 46 chromosomes, right? At the end of the S phase, like right here, just before the cell just is about to divide at some point, then we have 4 N chromosomes, or 92 in that case, right? So just for a very short while, and then the DNA is being divided into two cells. What we're going to look at today for this video is basically what happens here, right? At the molecular level, what happens to DNA and how is this DNA replicated? Okay, so this is what this video is all about. Now, let me just bring you back to the basics, something you already know. DNA, we represent this DNA as a double helix. We know that there are hydrogen bonds in between the nucleotides of each double helix, right? We know that. So it's those two double helix are, sticking together. The two strands of that one double helix are sticking together. And one thing that I haven't said yet is that the ends here of that DNA, the DNA is in an orientation that we call anti-parallel. Parallel. So what it means is that this is something that's parallel, right, on top of each other. However, one of the strands could be represented like this, while the other strand can be represented like that. So you see, they are parallel, but in opposite directions, right? Going this way here, going this way there. So basically, what I mean by that is, we have those two strands, but then there's a direction of all the molecules the way they are organized. The top strand is organized in one direction like this. And then the bottom strands, all the molecules are not organized like that. They are organized like this. So this is what we call anti-parallel, okay? So the molecules, basically, it's as if you were on a road looking in one direction, and on the other side of the road, people are going in the opposite direction, right? The road is parallel, but the direction is anti-parallel. So this is what DNA is. DNA is an anti-parallel molecule that happens to be a double helix. Now, to understand, because when we're talking about DNA, we're not talking about east, west, we're not talking about south, north, not talking about left and right. We're talking about a direction that doesn't matter where you are, the geography of it. There's no geography, okay? So we're talking about a molecule, and we need to understand, we need a convention to understand that we're always talking about the same thing. So it's easy to say, well, on the road you're driving, on the right or driving on the left, going in one direction, but for DNA it's not like that. However, the molecules that are interacting with DNA and the enzymes that are moving along the DNA, because they can move along the DNA, move in one direction the same way we move on the road in one direction. Okay, so that's important, and that's a rule that is always followed in one direction or another. So what I want to say now is that when it comes to DNA, and when we now know that there's an anti-parallel molecule, so let me forget about the helix part of it now, and let's just concentrate on the little pieces that are connecting this piece of DNA here, right? So we have the nucleotides in the middle here, and then we know that those nucleotides are of many, there's 4 different letters, 4 different molecules, and depending on the kind of molecule, they have either two hydrogen bonds, or 3 in the case of G's and C's. So AT, 2 hydrogen bonds, G's and C's, you have 3. And then it goes on and on. So I don't need to write everything here. What I want to say though is that in order to understand the direction of that DNA here, we use 2 numbers, and these numbers are 5 and 3 when we say 5 prime and then 3 prime. Now, why is it 5 prime or 3 prime? I'm not going to explain this in a video, but you will have access to this in your class material. And it's important for you to understand not so much about learning and understanding exactly what structure it is and being able to draw it from memory. This is not that important at this point. But what I want you to understand is why is there a 5 prime, 3 prime, and then understand that on the other side it's anti-parallel. So if we have 5 prime, 3 prime on the top, we will have 3 prime, 5 prime at the bottom. This is very important, okay? So I'm going to leave it there. I'm not going to explain more details about why it's called 5 prime, 3 prime. It's not important right now, because what we'll be doing is basically only talking about the DNA replication. However, I will use these 5 prime, 3 primes all the time during this lecture, because it's important to understand that when DNA is being replicated, it's done in one direction and one direction only for one strand, and then the other strand is the same thing. And this, as we will see, creates a set of problems for the cell. And it's kind of interesting to see how the cell deals with that, with these problems, okay? So it's a very good case of how to solve problems, okay? So anyway, I just want to keep it there, and then the next part that's just coming up, we will talk about some of those problems and what happens when the cell is in S phase and decides to duplicate its DNA, okay? Okay, so I decided to illustrate a DNA molecule with these tubes here, and you'll understand why I'm using this, because I think it's going to explain quite a few things, important things here. So these are my 2 strands of DNA. And one thing that I want to say here is that remember that these strands of DNA are are connected with one another via those hydrogen bonds, right? So it's not like they can go like this freely. They are connected, they are interacting with one another via the different bases and nucleotides that are interacting with one another on the two anti-parallel strands of DNA. And then these, so the DNA molecule, if it was not a double helix, if I say represent it to you in a two-dimensional configuration, it would look like that. So now the first thing that we need to do when we want to replicate DNA is that we need to separate those two strands, because the enzymes want to replicate DNA. So basically what they want to do is to, from one strand, like from one DNA like this, double-stranded DNA like this, they want to create two molecules of the same, right? Two of this exactly. So what we need to do is to use one of those strands as a template and copy the other one to make two molecules. So basically what we would have at the end of a cycle, at the end of the S phase, we start with this, but at the end we have two exact copies of this. Now what's interesting to note is this, okay? So that at the beginning we have two strands, and I'm illustrating this in blue, and those two strands are connected, and then when we copy them, when the cell copies that, what the cell does is use one strand right here, this strand right there, and it uses the other strand right here, right? So basically this DNA is being separated into two strands, and what it does, what the cell is going to do is to copy this strand here and include the other strand here that is complementary to this one here, A, T, G, C. So wherever there's a G, a C is going to be added. Wherever there's a T, an A T is going to be added. So you see here in red, right here, we have the new strands, right? The newly formed strand. But for each molecule here, the new molecules, we have the old strand that's from the template here. So those two strands were separated, and now they are incorporated in a new molecule. Okay? So that is an important part of that. You really need to remember that, right? Because that's important. It comes it gives you a better understanding of everything. So if you already know that, then that's easier for you to follow whatever is coming next. Okay? So now what I want to do is to show you that what happens with these two strands, right? So remember that we have this double-stranded DNA, and each of those strands is now going to be incorporated in the new molecules that we see here, here, and here, right? So we have two molecules. We started with one, now we have two. So what is going to happen, we need something that is a tool that basically is going to be able to remove all the hydrogen bondings between all those two strands, and that's not an easy feat because they're kind of like solid, right? A hydrogen bond is something that is not that easy to break when there's many of them. So what I want to show here is that what's going to happen is we're going to have an enzyme, and this enzyme is called helicase, and the helicase is going to get here in between the strand, it's going to bind to the strand, and then it's going to separate them. It's going to separate them like this, and what's going to happen is that some parts of that DNA will still be together, but then some other parts of the DNA will be like this. They're no longer interacting with one another. So you see the top strand here with the green tape on it, right? So that's the top strand, and then we have the other one at the bottom right there. And this here will happen all along the chromosome. Remember that each chromosome is a long stretch of DNA. So what we'll have is we'll have a lot of those features all along the chromosome. So we'll have one here, and then we'll have one somewhere else, and then we have one, over there. So if it can be created, there you go. So we'll have some of those. So along chromosomes, we'll have hundreds and thousands of these little openings like this. And each of those structures like that, there's a name for that, right? When we look at that DNA that is now being separated into two strands, it's going to create this structure right here. So let me continue on that here. and it's going to create this structure that kind of looks like this. And then we're going to have many of those structures along the chromosome, right? So these strands are no longer connected with one another. Basically, the G's are by themselves, the CZ is on the other side, etc., etc. So this part here happens because we have an enzyme here that's called helicase, and helicase is going in one direction, separating the strands. So this portion here is called a replication bubble. Replication bubble, okay? So this is our replication bubble, and on our chromosomes, linear chromosomes, long stretches of DNA, uninterrupted, we'll have many of those. So basically we'll have like those bubbles like this all over the place on the length of our chromosomes, right? So we'll have many different replication bubbles. Why not just one? Well, because DNA is long, we need to be efficient. The cell needs to be efficient. In order to be efficient, it just does that in many different steps. And it breaks it down so that it's easier and much faster to attack all of that molecule, this long stretch of DNA, at different spots, and then separate everything when everything needs to be separated. Okay, so it's just a question of efficiency. Now this brings a set of problems, and I'm going to show you what kind of problem this can create. I had no problems showing this to you because this is linear, right? But remember that when we're talking about DNA, we are talking about a molecule that is a double helix, right? So basically it is twisted like that, so as you can see right here. The problem is that if I do have a molecule like this that is double-stranded and also twisted as a helix, when I'm just about to replicate, I need to turn it on the other side. So what we have here is that we're going to have an enzyme, the helicase, that is supposed to undo the connections between the two strands, right? So here's what's going to happen. What's going to happen is that I'm going to use my thumb as a helicase, and I'm going to insert it right here. So I'm going to keep on twisting this molecule. Bear with me, it's not that easy. I don't have an assistant, so if I did, it would be easier. But let me just fiddle with this a second. And then, so what's going to happen is that we're going to, so I'm going to insert my thumb here. Obviously this is not going to work. So, okay, so I found a volunteer. I asked my friend Oscar here. Oscar doesn't speak much. He's kind of shy, but he's always ready to help. So take two for this part here. Couldn't do it without the help of my friend here. So there we go. So Oscar doesn't mind that I put my DNA molecule around his sternum. Doesn't hurt. Doesn't look like it, does it? Fine. Okay, so let's keep moving with this. So what I wanted to show is that once the helicase is supposed to do its work, basically separate the two strands like that, it needs to do so, but it doesn't do it on a molecule here that's on a board 2D, right? It seems easy to do this when you look at a board. However, now what we need to do is to do this in real life, and what we're dealing with is a molecule that has a 3D structure, but one of those things is that this molecule is twisted. So basically what we do is we have an enzyme that is inserting itself inside the bases and create a bubble, a replication bubble as we see here. As it does so, it moves in one direction this way, but also it moves in the other direction, right? So the bubble extends from both directions, not just one, but the other direction as well. I just drew one helicase here, but actually it goes in both directions. So this enzyme will open up the molecule here, and then here's what happens. You see, as I move my hand, you see, I pay attention to what's happening around here, okay? Let me bring this a little bit closer to you so that we can see it a bit better. So here's what happens. I have my molecule here, my helicase here, and it's moving, and it's moving, and it's moving, but look at what's happening around poor Oscar's sternum. You see there's more and more and more tension that is generated on that side here. Let me do this again, right? So let's look at this, and then you can tell that there's more and more and more, tension that's happening there. Now Oscar can't take it anymore, right? So what we need in real life, when you're at the molecular level, you need something that's going to help you relieve tension at this end right here. And that is the job of an enzyme called topoisomerase. So basically what you have is that when you have a molecule, a DNA molecule being open, you have the helicase, but in front of the helicase somewhere right here, you need another enzyme that's going to undo those twists, right? It's not going to do everything, but it's just going to undo enough so that it releases tension in order for the helicase to move through and be able to create those very, very important replication bubbles like that. Without those replication bubbles, there is no DNA copying that is possible. Okay, so that's a very important part. Okay, so let's just go back to this, okay? Just put it together so that we're not missing anything here. We have our two strands of DNA, they're antiparallel, we know that. And we always number these two, right? So 3', 5'. At this point, it doesn't matter which one is which, okay? We know the DNA is connected. And also, we know that we need to separate the strands in order to copy each of them, create two new DNA molecules made from part of the old molecule here, and then two new strands that are being added to the old strands. We already know that. Now, we know that there is an enzyme that's working its way on the DNA and then to separate the two strands. And we know that this generates problems because it creates tension. So we have another enzyme. We have one enzyme helicase, and we have another enzyme in front of it called topoisomerase that releases the tension. It's necessary. We've seen why that is. Okay? So now we have in our strands of DNA. So let me draw a long stretch of DNA here. And then what we know is that the opening of these replication bubbles happen at both ways, right? It happens, it goes this way, and also it goes this way. Basically, it spreads like that. Basically, it's just to go faster. So we will have a helicase at the front here, and another helicase there. So basically, and of course, topoisomerase in front of it right here, another topoisomerase right here. So one here, and then one here, working their way to help the DNA the separation of those two strands. Okay, so now that we know that, let's move on. So what we're going to have is these strands here, and then we will have several replication bubbles, hundreds, maybe even thousands of them, all along our chromosomes. So each of those are replication bubbles. And then what we have here is that we know this bubble is growing this way. Eventually, this is going to merge in a mega bubble, right? And we know that the bubble is actually growing from both sides like that. It's expanding, basically, right? With the correct enzymes, all right? So this part here creates some kind of a Y-shaped molecule. You see that there's a Y, right? It's like opening like that, right? So here, this is called the bubble right here. So we have the replication bubble, as we know. So we have replication bubbles, we have more than one here. And then what we have here in front of those bubbles, the expansion of that, that's called a replication fork. Rep fork. And of course, for each bubble, we have two replication forks, right? Because it goes in both directions. So what's going to happen is that those replication forks are moving towards each other right here. And then, of course, at some point, they're going to merge together like that, right? They are going to merge together. But at that time when they merge together, our DNA will be copied already. And we will see now what happens and how this DNA is being copied here. So you see, DNA is not separated all together and then copied. It separates and is being copied as it separates. It's being replicated as the DNA separates. It's not like we need those two strands. Okay, what do we do with them? Well, now we create a new strand. No, we separate them, but we also create our new strands as this DNA is being separated. Super efficient, but also super fast, right? It happens very, very quickly. So, but it's not without problems, and we will see why, and we will see how the cell deals with that. Okay, so this is the next segment of our of this video. Okay, so now that we know about replication bubbles, and that we know also that when DNA gets replicated, we keep bits of that old DNA. So again, DNA like this, our template DNA, will be used to copy that DNA, right? And then when that DNA is being copied, what we have basically is parts of the old DNA molecule here that are now being inserted in the new molecule, right? So we have these old bits here, and then we have the new DNA right here, this new DNA here that is now incorporated within the newly formed DNA here, right? So basically we start from one DNA molecule and then we have two, and then there's two new strands being added to that. Now, a few things that I haven't said earlier, and I should have probably, is that this DNA, when we copy it, we actually start right from a start point. And that start point is that a region of that DNA, and along our chromosomes we have many of those. They're not random, there are very specific sites here, and what we have here, they're called origin of replication. Okay, origin of replication, and this is where everything starts. So basically what happens with the origin of replication, the helicase can bind here and start to separate the strand. And remember that the bubble that is being created expands in both ways, right? So it forms those what we call replication forks, right? Remember the replication forks? So what we have here is that we have a bubble that's going to be created. Remember that now we know that we have origins of replication here. The helicase will forge ahead in both directions here. This is my triangle. My triangle here is the helicase, and then basically it opens up a fork here. So we have one fork right here. We have one fork right there. Okay? So now the origin of replication is where the replication starts, right? So that's important to understand. Now, let's go back to the basics of what the DNA molecule is and what it looks like. We know that we have a certain direction, 5', 3', right? We have our DNA in one direction, and then anti-parallel, so in the other position. Remember that this 3', 5' relates to the structure of the molecule. And if you look at it from the 3', so let's say look at a pen like this. So let's say that this is a 3' and let's say that this is a 3' and this is the 5' here. If I look at this from this way, the 3', well, it looks very different from that end, right? So this is the same strand, it has a direction 3', 5', but when you look at it from one end to the other, of course it looks very different. So that's how molecules are being recognized by enzymes. Enzymes look at the structure of some molecules, a 3' here, looking very different from there. And it's going to bind at some spot because it looks in a certain way, and not bind at the other spot because it simply does not recognize it. So this is an analogy that should help you understand what's coming up right now. So when we separate our DNA, we create this replication bubble, and of course the fork here. So what we're going to have here is a fork like that. And that fork is now the DNA that's being separated. And the DNA being separated is separated by the helicase that goes in this direction. So basically the helicase opens up, and don't forget that topoisomerase is in front and basically unwinds the DNA a little bit to allow the helicase to move ahead, right? Remember that part of the video. So this is here, what we have here in the middle. We still have our origins of replication, right? And then our DNA bubble, replication bubble here, is now this way with the helicase moving in this direction as well. Okay, so the helicase is moving in one direction, we have another one on the other direction. And remember that our DNA here is 5', and then our DNA on the other side at the bottom is 3'. So 5', 3', 5', 5', 3'. Okay, so that's important. Now, because it's called origin of replication, this is where everything starts. This is where this spot right here is where the magic happens. This is where our DNA is being replicated. So we have an enzyme that does that. Okay, so the enzyme is called DNA polymerase. I would write Pol, right? DNA polymerase 3. That's the enzyme that copies DNA. So basically what it does is that it starts here and it recognizes the 3', right? So remember, so the 3', 5', it will go in this direction. So it starts in the 3' direction, and it will go towards the 5' direction, right? So it will go from 3' to 5', and what's going to do is that it's going to, as it goes, it's going to add all those nucleotides and it's going to copy this new strand of DNA. Basically what it does, it's very easy, it just follows the helicase. It's behind the helicase and then starts to do its job, adding one nucleotide after the other. And this, of course, happens on both strands at the same time because we have the origin of replication. It opens up both strands at the same time. So our DNA polymerase 3 is going to be able to follow the helicase on the other side as well and add those nucleotides here. Now, one trick here that's important to understand is that it does follow direction 3' to 5', but what it does, it adds those nucleotides in a 5' to 3' fashion. Oh, wait, what? What did you just say? Yes, 3' to 5', it follows the template. But it adds from 5' to 3'. Yeah, why is that? Remember, DNA is an anti-parallel molecule. If we have 3', 5' on one side, then the other side, that's even if it's newly created, doesn't matter. We have 5', 3' on the other side, right? It's anti-parallel. It will be anti-parallel both ways. Anti-parallel here, anti-parallel here, anti-parallel here. So what we have here, again, is 5' to 3', right? It goes in this direction and this direction here. Now, it's easy because it follows that helicase. Now, can you see a problem here that is being generated, right? Just think about for 2 seconds what problem that is, okay? That problem is that the enzyme can polymerize, add nucleotides from 5' to 3'. That's what it does, right? It adds from 5' to 3'. That's what it does. Now, in order to do so, it follows 3', 5'. Hopefully I'm not confusing you here. You need to think about that. Now, it reads in 3', 5' fashion, but if we want to duplicate this DNA at the top here, you see we have a problem because it needs to do it in this direction, right? So what it does is that basically it will copy this in this direction, but this helicase is moving along. So what it means is that at some point, we will need to interrupt this synthesis. It will start right here and then move along until it meets this part right here, no problem there. But then the helicase at some point will be further down, so it needs to start again a bit further. So you see that the synthesis here, right here at the bottom, is continuous. Continuous, right? It keeps on going, but this part here is always interrupted because it needs to follow the helicase, and then, but it's in the wrong direction. Same enzyme, DNA polymerase 3, but the DNA polymerase 3 does only synthesis from 5, it adds from 5 prime to 3 prime. That's what it does. So it reads 3 prime to 5 prime. Well, it can't read it here because this here is always, it's not separated yet. So what do we do, right? Well, we wait. We wait until the fork opens again. So here's what's going to happen after a while. What's going to happen is that we're going to have our DNA molecule, and our DNA molecule will be like this, right? Will be like this. We have our replication forks all over the place. And then we will add those DNA polymerase 3. It's easy for it to create this part right here. And it's very easy because what it needs to do, starting from the origin replication here that's in the middle, don't forget, I'm not going to draw it, but just remember that. And then the helicase moves this way. So this here can be synthesized following the helicase here. Not a problem at all. Not a problem. But what we need to do here is that we need to create those little pieces here, and it can only be created as the DNA is being opened. So we basically have a strand that is non-continuously created, and that, because of that, we say here that there is a leading strand right here, this part here. But also here, because it's being interrupted all the time, this will be called a lagging strand. Okay? Leading strand, lagging strand, because of the 3', 5' nature of those nucleotides. So that's very important. Okay? So, this creates problems for the cell. One goes in one direction, but we need the other one, we need to always go back. Of course, the cell has a solution for this. It has evolved over millions and billions of years to do something like that, so it's very good at it. But, when you look at it, it creates obviously a problem. So this is why you'll see leading strand versus lagging strand. Okay, now what we're going to do is basically look at the specifics of how the strands are now being created. Okay, so I just want to go more to focus more on that part here and look at the molecules, what exactly happens at that part. Okay, so let's look at the details. Okay, we have our 2 strands of DNA in blue, the strands that were originally in the same molecule. They are separated in a bubble. This is what we have here is the focus on the bubble, 3', 5', 5', 3'. The top one, that is what we call the leading strand, leading strand. And it's called a leading strand because basically it's following the fork, right? So the fork goes this way, it's following the fork. Now the problem is that on that side right here, if we focus just on that side here, the enzyme, which is DNA polymerase 3, can only synthesize from 5' to 3'. So it reads in 3', 5' fashion, but it adds 5' to 3' on the antiparallel strand. So because the fork is moving this way, on the other side we have a lagging strand because the DNA polymerase cannot follow the helicase. So basically it needs to backtrack all the time. What a problem, right? So in order to do this, we still have the same enzyme. It's still DNA polymerase 3, but it's a bit more complicated. So what we need is a small enzyme. Is an enzyme that's going to add a small small piece of nucleotide. And that's not small piece of nucleotide is going to be RNA. Okay, RNA. So what's going to happen is that an enzyme called primase is going to prime the DNA to be, for the RNA, the DNA polymerase 3 to interact with it. So what it's going to do is that it's going to add a small piece of RNA right here. Basically, it's kind of like, see it as an anchor for DNA polymerase 3. So if there's no anchor, DNA polymerase 3 does not know where to start. But with this primer, the DNA polymerase can now start here. Again, don't forget the fork is moving this way. This is a piece of DNA that was here synthesized prior. So you see it's synthesized, moved this way, but it's synthesized backwards in a way. And now what's going to happen is that we're going to have the DNA polymerase 3 that is going to be now able to recognize this part here, and is now going to add these nucleotides here, right? Again, from 5' to 3', because it's following in the other direction, right? So it's going to go in this direction right now, okay? And then it can do so because it has a little primer here. The primer, not prime, but primer, is added by an enzyme called primase. Primase. And then what's going to happen is that all along this DNA, as it moves, we're going to have our molecule that is going to be done like that. So what we're going to have is 3', 5, 3', 5', 3' here. What we're going to have here is our molecule, newly formed molecule here, by DNA polymerase, following the replication fork, not a problem, leading strand. But what's going to happen is that we're going to have a bunch of small pieces of DNA here being synthesized one after the other on the lagging strand. And some of them, actually, each of those little strands here have a little piece of RNA here at the top, okay? So you see, we have pieces of DNA, RNA at the top here. Now, in order to do this, we're going to need to complete all those links in between. We're going to need a new enzyme for that. Okay, so we'll need it because there's pieces missing here. There's nucleotides missing in between here. We'll need another enzyme, and that enzyme is going to move this way here. It's going to move in the opposite direction as polymerase 3. Polymerase 3 move this way. Polymerase 1 move the other way. And what it's going to do, DNA Pol 1, will move in this way here. It's going to remove those little primers and then finish the last strands, right? So it's going to finish everything here. But however, it leaves a little hole in the middle here. It leaves a little hole because it cannot complete the link between the last nucleotide here and the first one here. You see, there's a covalent bond that is needed here, right here, right here. So it's not, the DNA polymerase cannot do this. So there's another enzyme, and that other enzyme is called ligase. Ligase. So basically what's going to happen is this. If I can just erase this now, and then look at what's happening, we have our pieces of DNA that are partially created like this. Right? And then we have our full part of DNA here at the top, and all are paired with nucleotides. What we had is a bunch of these here were added by Pol 3, and then because there was a gap, those gaps were filled by DNA polymerase 1, and each of those small fragments here are called Okazaki fragments, Okazaki fragments. So the small pieces of DNA are called Okazaki fragments. And then at the end, DNA polymerase has done its job. Now what it needs to do is fill this gap here. And it can't do it because it's not made for that. So what we need is another enzyme right here that's going to fill this gap right here. And that gap is filled by ligase. Okay, that's the enzyme that does that. So now hopefully what you understand from that video here is that what we do is we need a replication bubble. The replication bubble moves in both directions. The enzyme starts at the origin of replication, and on one direction they move following the helicase. But because both strands need to be formed on both sides of the origin of replication, we need the enzyme on one side that was going to create a leading strand following the helicase, but on the other side, it will have to backtrack all the time in order to follow the helicase. But because the helicase move, and because the DNA polymerase functions in the other direction, it needs to go back all the time. So it's the same enzyme polymerase 3 that does that, but it just does that in reverse in a way, in the opposite direction to the original replication. Hopefully that makes sense. But it can do so only with the help of other enzymes. So primase is very important because it adds a little anchor on which the DNA polymerase 3 says, okay, now I can sit here, I can do my job, so that's good. But then this creates a hybrid molecule with pieces of DNA and a tiny bit of RNA that needs to be removed. So DNA polymerase 1 is going to cover all of those nucleotides that were occupied by an RNA. It's going to remove those, put new DNA nucleotides there, but then at some point it's going to leave a gap. That gap will need to be filled by another enzyme called ligase. Okay, ligase is going to create that gap. But in the end, all of this community of enzymes are working together to copy both DNA molecules, and that's what we want, right? That works very well this way, but it's just a bit more complicated. Now, I posted in the important materials, I posted a few videos, and it's very important for you to watch those videos because it explains this with a very, very amazing animation, and I really want you to look at that because that's very important. So please look at this and understand all of these enzymes work together in order to copy DNA. Okay, so hopefully this is much clearer for you, and you understand now what DNA replication is all about. Thank you.